Hi class, this is your lecture, the first of two on the kidneys and the renal system. You'll have to bear with me a little bit. I misplaced my glasses, so I might be moving back and forth a little bit to see the screen. I apologize. All right, we are going to go through most of the chapter. We'll talk about what excretion is, but otherwise we'll focus on sections 19.1 through 19.6. All right, so let's talk about just the general functions of the kidneys, and then we'll move beyond that and talk about the function of the renal system. But the kidneys are important in maintaining and helping our body to regulate the fluid volume, and that does tie to our blood pressure. They're also really important in maintaining the osmolarity of our body compartments. They're important in maintaining the balance of the various ions. And of course, we know that ties into osmolarity. The kidneys have a very important role in helping with the regulation of our pH, and they can help to correct when there's problems there. And then they're involved in the removal of metabolic waste products from our bodies. They are associated with the production of some hormones, and we'll learn about a few of these here. And then we'll also talk about them in our last section of the semester, which deals with endocrinology. Okay, so the functional unit of the kidney is called the nephron, and there are millions of nephrons within each of our kidneys. Now, each nephron, or the kidney itself, and the nephrons have two layers to them. So the outer part is called the cortex of the kidney, and this is where about 80% of the nephrons are found. These are the common cortical nephrons. The other 20% of the nephrons are in the inner part of the kidney, in the medulla. And these are called the juxtamedullary nephrons, since they are next to the medulla. Now, our kidneys are very vascularized. In fact, they are one of the places in our body where we have a portal system, which is two successive capillary beds. And you know what capillary beds are for. That's all about exchange. And that's what the kidneys need them for too, to exchange the various materials so that the kidney can perform all of its jobs, including maintaining pH and osmolarity. The path of blood through the kidney comes in through the afferent arteriole. It then goes to a specialized area of the nephron called the glomerulus, and then it goes out through the efferent arteriole to the peritubular capillaries known as the vasa recta. So we have a dedicated portal system associated with our kidneys, with each kidney. Okay, so now just looking at an overflow of the urinary system, I'd like to point out the kidneys are up high. I mean, they're actually about here on us. They are protected underneath our rib cage, at the very base of the rib cage. A lot of times people will mistake pain down here in their lower back or buttocks as being kidney pain, but that's not where the kidneys are located. They are a vital organ, and like most of our vital organs, they are protected within a structure, in this case, the rib cage, as well as how tightly positioned everything is within the abdominal cavity. So we have our two kidneys, and then they have a urethra that goes to the urinary bladder, leading to a single urethra. And then looking over on the right side of it, you can just kind of see the base of the ribs shown here. So they're really just right underneath that lower rib area. Look how gigantic these blood vessels are. And that ties to the function of the kidneys. We have the inferior vena cava, as well as major vessels. This is the aorta. Now, looking at a cross section of the kidney, so if we're to cut it in half, this is what we would see. So notice there are different colors to indicate that there are different structures, and different structures are associated with different functions. So the cortex, this outer lighter color, is where 80% of the nephrons are. And then in the medulla is where we will see the juxtamedullary nephrons. I'll point those out to you in a moment. But you can see that the kidney is organized into repeating units. We've got the same basic structure over and over again. And then this is an encapsulated organ. So there's a capsule around it. Now let's look over at this portion of the image, part D. So you can see some of these nephrons right here. This tan structure is a nephron, and this would be a cortical nephron because it's located in the cortex. The other nephron here is one of those juxtamedullary nephrons, in that it goes into the medulla, but parts of it are up in the cortex too. So I don't want you to think that the juxtamedullary nephrons are all in the medulla. They're not. They span both areas of the kidney. You can see each of these lobes of the medulla is very well vascularized to support the functions of the kidney. Okay. All right. So this is a little bit easier to appreciate on a bigger structure. So this is the cortical nephron, and then the one beside it is you can see it's dipping down into the medulla. Okay, we've got our blood vessels, one of them being the afferent arterial and then the efferent arterial, going into each nephron. So every nephron has a blood supply to and from it. Now look at part F of the figure here. What it is showing you is that each nephron is highly associated with blood vessels. And this is so that we can have this exchange of materials. Our body's very efficient at recovering materials out of what will become our urine if we need them. So that way, for example, glucose normally isn't released in the urine. It is brought back into the body at a certain point in the kidney so that it can be utilized again. So our body's very efficient at getting back the materials it needs and releasing the metabolic waste products. And that's accomplished by having all of these very extensive peritubular capillaries. They are wrapping around the tubule of the nephron. So part H is showing you one of those juxtamedullary nephrons. It too is associated with the vasa recta, and both types of nephron drain into what is called the collecting duct, which then eventually feeds to the bladder. Multiple nephrons feed into the same collecting duct. So you can think of it as a merge back in to these common structures. Now, a kidney contains about 1 million nephrons just in one kidney. And then of course, we've got two. People can live quite well with just one kidney. All right, so now we're looking at a simplified nephron, and this allows us to appreciate the different structures of it. And you do need to understand what these different structures are because they have different functions. And certain material can move at certain places in the nephron. For example, water can move in some places where ions can move in other places. So the structure is very much tied to the function here. So this section up top, this kind of ball part, is the all of it. It makes up, well, the blood part of it is the glomerulus, and then the tubule part of it, the nephron part of it, is called Bowman's capsule. And these together make up the structure. Now, Bowman's capsule next feeds into what is called the proximal tubule. That means adjacent or next door. And there's a lot of folding of it. And then the proximal tubule descends into the descending loop of Henle. And then we have an ascending loop of Henle, which then feeds to the distal tubule. And then finally to the collecting duct, which goes to the bladder. Multiple nephrons will merge into the same collecting duct as a point to feed the material into the bladder. So the main sections of the nephron are the glomerulus surrounded with Bowman's capsule, and then we have the proximal tubule, the distal tubule, the loop of Henle with its descending and ascending limbs, and then finally the collecting duct. This picture here is showing the blood vessels that form up this glomerulus. So this is one of the capillary beds associated with the portal system of the nephron, the glomerulus itself. And these are areas that can be damaged from high blood pressure. This is a filtration unit. So anything that clogs up the filtration ability could have an impact on the body. So in my research, when I was at the University of Arizona, I studied this section of the kidney because I was interested in learning more about the autoimmune disease lupus. And this is one of the parts of the kidney that is affected by lupus. And we actually found some really interesting things. We found viruses present in the glomerulus that were associated with people having more severe disease. Okay, so the function of the kidney or of the nephron, it is to filter, that's part of it, but reabsorption, secretion, and then finally excretion are also critical functions of that nephron. So excretion is what goes in the toilet in the form of urine. We're not going to spend time talking about that, but we do need to understand how to determine what is called the filtration fraction. So filtration comes from the blood going in through that afferent arterial into the glomerulus, into the nephron. So the structure of the glomerulus with the tubule, Bowman's capsule around it, that is what is called the renal corpuscle, that whole structure. So it contains blood vessel and tubule. Material that moves through this section into the glom is called filtrate. And it is destined to go to the urine unless material is reabsorbed out of the filtrate. And most material is reabsorbed out of that filtrate. So that brings us to the second function of the nephron and kidneys, where material has the opportunity to go back into the bloodstream. And this is happening with those peritubular capillaries that wrap around the nephron. So reabsorption is a very important part of our renal system. Anything that is not reabsorbed back to the bloodstream ends up in the urine. So reabsorption is very directional. It goes from the filtrate to the bloodstream. It is not the other way around. Secretion's the other way around, going from the bloodstream into the filtrate. And that happens in the kidney too. And it is also in association with the peritubular capillaries. So we've got our filtration, reabsorption, excretion, and then what is left over in the filtrate is excreted in the form of urine. Okay, so now let's look at the nephron, and we can apply these different functions to the correct locations here. So we have our efferent arteriole feeding blood into the glomerulus. So this is one of the capillary beds associated with the portal system. And everything that goes through out or passes through the epithelial layers of the glomerulus is what becomes filtrate. And then material, the blood goes right back out, the afferent arteriole, and then feeds the peritubular capillaries. So this is definitely meeting the definition of a portal system in that we have these capillary beds that are connected together. So filtration happens in the corpuscle. Reabsorption can happen right away in the proximal tubule. Secretion can also happen here. As we move down the descending loop of Henle, it's all about reabsorption. That is the function of the loop of Henle, is to provide an opportunity for our body to reabsorb materials, ions, glucose that would exit the body. This way we don't need to make more of it. For example, potassium is reabsorbed here along with our other ions. So as we move up the ascending loop of Henle, finally to the distal tubule, this is also an area where there's reabsorption and secretion. And then finally, the same is true in the collecting duct. And then everything else that's left in the filtrate that was not reabsorbed, that was secreted, goes to the bladder. Now, it is possible to recover fluid out of the bladder in cases of severe dehydration, but that's not normal physiology. So typically when our filtrate makes it in the form of urine into the bladder, its destination is out of the body. But I would be remiss to tell you that it can't be recovered because it can under extreme conditions. Now, what we are going to learn throughout the rest of this chapter and into the next chapter, chapter 20, is that different materials are reabsorbed at different places along the length of the nephron. All right, so here's a table that just describes the different sections of the nephron and lists their functions. Okay. So now let's dig a little bit deeper. In some ways, this is a more simpler figure, but we're focusing on the blood flow coming into the glomerulus and then right back out the afferent arteriole. Whatever passes through in the filtrate then moves into the nephron, where it is either reabsorbed to the bloodstream or material is secreted from the bloodstream into the tubule, and then whatever is left over is excreted. So we can figure out the amount of solute excreted by knowing the amount filtered minus what goes back to the bloodstream plus what comes into the filtrate is the formula to calculate the concentration of a certain solute that is excreted. So the excretion of every substance, every ion, glucose, is dependent upon filtration, reabsorption, and secretion. So the percentage of filtrate that moves through into the tubule is the filtration fraction. And we need to consider both the blood vessels, the capillaries of the glomerulus, plus the Bowman's capsule itself, because both structures are needed in order for filtration to happen. Now, we've got filtration occurring at a few places, and we also have a number of factors that can impact filtration. One of those would be the endothelial layers of the glomerulus. If material is going to filter out of the blood, it needs to pass out of the blood vessel. So that's one layer of filtration let's call it a filtration barrier that has to be overcome. The kidney, the nephrons the vessels have the fenestrated capillaries, which are those bigger pores. So that's helpful. Material would also need to move through the basement membrane, and additionally through the epithelial layer of the capsule itself so that it can enter the tubule. Here we've got what are called podocytes. We'll talk about those, but you can see from the name, pod means foot, so a foot cell, if you will. And these are it's an interesting anatomy. These podocytes are able to move, and then they can come and cover up the holes from the fenestrated capillaries, preventing movement. And then we've got the mesangial cells as well that are present in that epithelial layer of the capsule. Okay, so let's take a look. Let's put some numbers in it now. So the material that comes into Bowman's capsule and the glomerulus, 80% of it goes back out the efferent arteriole. So it's only about 20% of the volume that goes into the kidney moves into the tubule. So the majority of it is going right back out the arteriole, but this provides opportunity for a lot of that blood flow and interaction to happen so that there is efficient filtration. And then moving down the length of the nephron, most everything is reabsorbed. About 19% of the fluid is reabsorbed back into the body. So we're recovering about 99% of the plasma that is associated with moving through the kidney. That leaves us with less than 1% of the volume that is excreted into the environment in the form of urine. Our kidneys are efficient, as we've seen with many of our other organs and organ systems. All right, so now let's take a look at how these different filtration barriers work. So all of this is the corpuscle. We've got the glomerulus and then the Bowman's capsule. These purple structures that are wrapping around the vessels of the glom, well, these are the podocytes, and they can actually move. They move, so you can think of like a spaghetti colander that you use to make pasta. Well, if you have something on the other side of that colander blocking the holes, you're not going to get very efficient filtration. So the podocytes are tied with the amount of material that filters through. So they definitely are a barrier that is needed to be bypassed. Now we've got some different, just notice the colors here around the nephron. We've got these two different areas that are tied to helping to regulate some of the function in the kidney. I'll come to those in a moment. We can actually see an electron micrograph showing the podocyte, and look at how extensive the surface area is of that podocyte. All of these structures, even these little ones, are part of that podocyte, wrapping around the blood vessels. Okay. So the purple, again, is the podocyte, and we've got one capillary shown here to keep it simple. This is the glom. Here's the mesangial cell, which really is helping to space the different materials apart and helping to keep everything together at the same time. And then we've got the podocytes stretching around. You can see them all around the blood vessels. So let's take and zoom in a little bit at this section. So in order for the material to filter through, there has to be an opening. So these podocytes can cover up those filtration openings or not. So over in this part of the figure, for example, you can see that there is an opening in between these two podocytes. So this is a filtration slit that allows the material to go through our various layers to make it into what is now properly termed the filtrate, which would be inside of Bowman's capsule. Okay, now pressure is tied to any type of flow, any type of movement as we've learned, but let's identify some of the different factors that affect what is called the glomerular filtration rate. That's what GFR is. So, okay, the main driving force here is the blood pressure coming into the glom. So our pressure at this point is about 55 millimeters of mercury. But we have two opposing forces that are subtracted out, if you will. So we have the pressure, this 30 milligrams of mercury here. This is the colloidal osmotic pressure, and it's due to the proteins that are present in the filtrate. But we need to subtract that 55 minus 30. And then we have this other 15 millimeters of mercury that needs to be subtracted too. And this is the pressure created by the fluid moving in Bowman's capsule. So once we take out the two opposing pressure forces, it leaves us with a net filtration pressure of about 10 millimeters of mercury. And that is sufficient to do the job. The kidney is designed to work optimally at this pressure. So one of the problems that can happen in a person that has untreated high blood pressure is that they can permanently damage their kidneys because the pressure going through is too high. It does take time for that to happen. So this is another reason to advocate for treating blood pressure before it creates problems. So we have a couple different ways that we can calculate the net filtration pressure. We just went through this formula on the top where we take the blood pressure and subtract the colloidal osmotic pressure as well as the pressure from Bowman's capsule. But we can also look at it a different way. So this is an alternate formula that we have in the lab so that you can calculate it both ways. But it's basically the same thing. It's the blood pressure minus those opposing forces. Okay, now our body is actually really good at dealing with changes or quite a large range of pressure changes in the kidney. And this helps to prevent damage. So take a look at this. So we've got a graph here showing the arterial pressure along the x-axis versus the glomerular filtration rate along the y-axis. And what it shows us is that our body is able to deal with a range of pressure, quite a range from about 80 to 180 millimeters of mercury without having an impact on the kidney. And that's due to the regulatory processes that are in place in the kidney. So it can deal, autoregulation is capable of correcting the pressure going through the kidney over this large range. Let's see how that's done. Okay, our top figure here, we're looking at, we've got our arterial. So notice that it's not clamped down yet. So we've got blood flow to other organs. We've got blood flow into the glomerulus, which is the main driving force of the filtration rate. But what happens if the arterial, that afferent arterial, clamps down, if it constricts? Well, then we push more blood to the other organs, and we have less going into the kidney. So that would result in a decreased glomerular filtration rate, since there is not as much force to push it through in the first place. So we see that vasoconstriction, right? We clamp down, that increases resistance, but it decreases flow through the kidney and results in a lower GFR. In the bottom right side of the figure, we're seeing the resistance applied on the efferent vessel. So we have we've got more shunting of the blood going to other organs due to the clamping of this efferent arterial, but the blood is going through. And in fact, since it's not clamped down until we're at that efferent exit, it results in an increased filtration rate. We have decreased blood flow, but it's with a higher filtration rate. All right, so let's look at how some of this regulation happens. So this is called tubular glomerular feedback, talking about the structures, right? The tubule and the glom. Okay, before we go through the steps, I would like to point out some of these different structures that I referred to earlier. We've got this one area called the macula densa, and it is similar to how our barioreceptors work, in that it's always sensing the flow through it, and that allows it to tie to this regulatory process. So as we have an increase in the GFR for whatever reason, that means there's more blood flowing through the tubule. We're pushing more through because we've got an increase in our filtration rate. So what that does is it drives more blood past this macula densa, and the macula densa is able to sense the flow. What happens then is a paracrine signal is released from the macula densa that acts upon the afferent arterial going into the blood, into the glom. And then that causes vasoconstriction of that efferent arterial, which then will help to decrease the filtration rate, getting us back to normal. So the end outcome is that the glomerular filtration rate decreases due to this autoregulation. So keep in mind that part of the figure is just missing here. It's just not showing the loop of Henle, but it's not disappearing to nothing. All right. Now, we do have a number of different mechanisms that material is able to be reabsorbed to our body. So that is going from the tubule back through the epithelium, through the extracellular fluid, and then finally back to a blood vessel. But different materials move through different mechanisms. We've already learned about some of the different channels and pumps that are utilized to move our various ions. Well, sodium, the sodium ion, it is moved through a pump, through active transport, to get it to go back to the extracellular fluid, to the bloodstream, because we're going against its concentration gradient. We have the movement of different ions. Some of them are able to go through both epithelial layers of the tubule, where others squeeze through an opening. We've got water, which is going to move through osmosis, but some of it can go directly through. I mean, water does move on its own through the cellular membrane. It just doesn't go quick enough to support life. That's why we need our facilitated diffusion of water. But once water's moving, that will start to affect the concentration of other solutes in both of these locations. And then we've got some other material that can pass. So we'll go over some of these newer pumps that we haven't learned about yet. But you know that urea is a non-penetrating solute, so it does cross and will equilibrate under the right conditions. So filtrate is actually really similar in composition to the interstitial fluid because of the movement of these ions right over to that extracellular fluid. Okay. Now, most our organic materials are going to be absorbed right away in that proximal tubule. So that's the first part of the tubule we get to after moving out of the corpuscle. So let's go through and take a look at some of our old friends here. We've seen our pump here before. So what happens now is we've got a gradient of sodium in the filtrate, in the tubule. And the sodium is able to move using a variety of different membrane proteins that follow the gradient. So we've got the movement of sodium in, and sodium goes out the sodium pump. And in exchange, with the help of energy, we're pumping our potassium back in against its gradient too. So this is the old friend, our sodium potassium pump. And this is active on that basolateral side, on the side of the interstitial fluid. All right, we'll look at the sodium glucose cotransport. So we've got a similar setup. We've got a high concentration of sodium ion in the filtrate, and we do have some glucose. It's at a lower concentration. The sodium glucose transporter is this here. So glucose moves in and it co-transports, sodium moves in actually, and it brings in glucose. So glucose is able to move through that epithelial cell and then out another transporter back into the extracellular environment. Sodium is going to run through the sodium pump and bring potassium back in. And then of course, we've got our other leak channels that we learned about earlier in the semester. So sodium, following its gradient, is able to pull glucose into the cell against its concentration gradient. And then the glucose diffuses out that basolateral side, allowing the sodium to take action through that sodium potassium pump. So glucose transport is dependent on the sodium gradient here. Looking at urea, so urea is, as we know, is a penetrating solute. So it will move, and if the circumstances allow it, it's going to reach equilibrium. But in the kidney, things are constantly moving. It's a continuous process. So we don't. Well, just keep in mind it's a continuous process. So to begin with, the concentration of urea in the filtrate is about equal in concentration to what is in the extracellular environment. Keep in mind that the composition of the filtrate is really similar to the extracellular environment. That means we don't have the gradients established to move many of our materials through diffusion. So we have to have other ways to do it. So what happens is once the sodium goes through and the other ions and solutes are reabsorbed, water is going to flow because of osmosis. And then that is what affects the urea concentration. Once water moves out, the urea concentration is going to increase in the filtrate, and now we have a gradient and it can diffuse out. All right, let's look at the secondary transport of glucose here. So we're in the proximal tubule again. It's where we've been the whole time in these last few examples. Sodium moves in following its electrochemical gradient into the sodium glucose transporter. So sodium in, I keep saying that. Sodium moves in and it pulls glucose in. Excuse me. I'm having a hard time seeing because I lost my glasses today. So our glucose will go out on the other side through the protein. And then sodium, again, goes through the sodium-potassium pump in exchange with potassium. So we have examples of active transport, directly harnessing that ATP, the sodium-potassium pump. We have examples of secondary active transport happening with this glucose-sodium transporter protein. And then, of course, we've got facilitated diffusion of glucose through its diffusion channel. So different material moves through different mechanisms. That's really no surprise to us at this point. Okay, now this is a good time to talk about what's called the renal threshold. So I'll give you a clinical example, and we'll see this in the lab this week or next week when you do your urinalysis. Most people don't have glucose in their urine. Why? Is it because there's no glucose in the filtrate? No, we just saw that there's glucose in the filtrate. Most people don't have glucose in their urine because the body is efficient at getting it back, at reabsorbing it back. But there is a threshold to the amount of material that can be reabsorbed back to the body. That's the renal threshold. And it simply is the concentration of a substrate in the plasma when we're at maximum transport. So in the graph, we're looking at the plasma concentration of a substrate that is increasing in concentration. This is on the x-axis. On the y-axis, we're looking at the transport rate of that substrate. So just substrate X, if you will. So what happens is we are able to transport the substrate X up to the point when all of the transporters are saturated. And at that point, we can't transport anymore, regardless of how much is there, because everything is saturated. So think back to when we learned about our receptors. If all the receptors are bound, then there's nothing that can be done. So the reason why people with diabetes will sometimes have glucose in their urine is because they have so much glucose in their body that is filtered into the filtrate that they reach saturation. And then whatever's left over can't get reabsorbed. So that means it stays in the filtrate and is excreted in the form of urine. So different materials have different thresholds. And it's all tied to saturation of the various transporters that are used in moving this material. So let's look at the example for glucose. We're actually looking at the same graph here. It's just broken up so that we can break it down by the different functions of the kidney, namely filtration, reabsorption, and then excretion. So in the top left, we're looking at filtration. We're looking at plasma glucose levels increasing on the x-axis, and then the filtration rate on the y, and we are able to filter it in direct proportion to the concentration of plasma glucose. So filtration of glucose is proportional to the plasma concentration. The filtration part is not what saturates, right? The saturation part affects reabsorption. So now let's move to reabsorption. So we've got our same setup on the x-axis, but on y, we're looking at the reabsorption rate. So as we increase the concentration of plasma glucose up to the transport maximum of 375 milligrams per minute. I think that's what it says. I'm sorry if my numbers are off. I can't read it that well. But we get up to that transport maximum and then it levels off. And at that point, so over 300, it would be present in the urine. The glucose would be present in the urine. So this purple range is our normal physiological range for glucose. And you can see that we, under homeostasis in our normal physiology, we are able to recover more than our normal physiological range. So that means that even after we eat a big meal or have a bunch of candy, we still have some wiggle room on the other side to deal with that, to deal with the increased sugar from that. However, a person with diabetes, though, everything's going to be shifted more over because they have so much more to begin with. So the reabsorption of glucose is proportional to its plasma concentration up to saturation. Now, excretion is the filtration, the material that is filtered, minus what is reabsorbed. So for glucose, it's going to be everything on the other side of the transport maximum, which is 300 mg per 100 ml plasma. So glucose excretion is 0 until that renal threshold is hit. And now let's put everything together. This bottom graph is a composite that shows filtration, reabsorption, and excretion of glucose. So we are able to reabsorb glucose up to our transport maximum, which is normally adequate for most of us when we're in our physiological range. But in the circumstance where we have more glucose than our transport maximum, that is where we see excretion of glucose, beginning at the concentration that exceeds that maximum threshold for glucose. So I'm going to set up the lab next week so that we can look at the class data and have a data set to compare. And we should see that most of us do not have glucose in our urine. All right, now let's apply this back to the pressure, and the pressure gradient or circumstances do favor the reabsorption of materials back to our interstitial fluid, or back to the bloodstream eventually. And so why is that? Well, let's look at our pressure dynamics again. The blood pressure, filtrate pressure is 55, but we have to minus out our opposing forces that come from the proteins that are present, as well as the pressure created in Bowman's capsule. So we're left with a net driving pressure of 10 millimeters of mercury going down into the proximal tubule. Now, the proximal tubule, we've learned, is wrapped, all of the tubule, the nephron, is wrapped with the peritubular capillaries, but the directionality favors the movement of material back to our bloodstream. Let's see what I mean. Okay, so we've got our 10 millimeters of mercury coming down, and then we've got pressure in our blood still, right? We've still got our peritubular capillary pressure of 10 millimeters of mercury, but our osmotic pressure in the blood is about 30. So at this part of the kidney, we've got a pressure gradient of 20 that is favoring reabsorption back to the bloodstream, back into the peritubular capillary. So, you can think of it as a way to draw the material back to the bloodstream because the pressure dynamics support that to happen. All right, so secretion is when material goes from the extracellular fluid into the nephron. So we're getting rid of it. It's being destined to become urine. But this is important we need to talk about the role of pH balance, and that does tie in with the role of potassium. Potassium balance and pH balance are tied together because of some of the pumps. Okay, I didn't mean to jump off of that so quickly. So we will talk about the rest of this part of it in the next chapter, and we'll look at examples of what happens with altering concentrations of potassium. Okay, so that does finish us up for chapter 19. I'll be back soon with your chapter 20 lecture, and I hope everybody's doing well. Take care. Bye-bye.